Yo, 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 welcome to Gene Regulations in Eukaryotes. Just like prokaryotic cells, eukaryotic cells must also regulate expression of their genes. We know this because, again, all cells in an organism contain the entire genome, but not all cells look alike for that multicellular organism. For example, we don't have any heart cells in our nerve cells and vice versa. So again, we need to know what genes are turned on and turned off at the right time in the right place. And so the differences in gene expression between cells is what leads to differentiated cells or specialized cells. And it results in different cell types. We can see the start of it with the stem cells. And then we can find that differentiation in the next step, and then in the organ where those cells are found. So again, like I said, we can't find any nerve cells in the hearts, and then we don't have any cardiac in the brain. They're exclusively to their own path. So in order to understand eukaryotic gene expression, we must dive into how these DNA would look on the molecular level, right? So we know the DNA at the very base of it has sequences of nucleotides here, and then when we zoom out, this structure is called the chromatin, okay? The chromatin. Very compact DNA protein complex. And so within the chromatin, DNA is wound. You can see that there, the string wound around groups of 8 protein molecules called the histones. So the histones are these guys. And then the DNA looks like a string around the beads. So each bead is considered to be a nucleosome, and we see the labeling here. Protruding out of the nucleosome, which will be important later on, would be these histone tails. And so the levels of chromatin packing are determined by the degree of nucleosome coiling. And we see this zoomed out picture of the chromatin, and then each one of these spheres would be the nucleosomes. So again, describing the chromatin structure, we have regions of the chromosome where these chromatins are loosely coiled and it's called euchromatin, and then tightly packed chromatin called heterochromatin. So their definition tells us a lot about their transcription ability. So if they're loosely coiled, it means that transcription can occur because there's room for the RNA polymerase to bind to that DNA. So we have euchromatin being that. And again, one way I remember it is that U is usually true. And so true chromatin DNA is used to transcribe. So boom, there we have it. It is being transcribed by RNA polymerase. Now, the other one is tightly packed, which is heterochromatin. So sort of chromatin. It's chromatin, but sort of, and it's transcriptionally inactive. Because it's tight, there's no room for that RNA polymerase to go in and transcribe it properly. So modifications to the histones and DNA of chromatin influence both the structure of chromatin as well as gene expression. So the first process we're going to take a look at is called histone acetylation. So acetylation means that the tails, those protrusions I mentioned earlier, are acetylated. Acetyl group is added, so that is the CH3 group. And so again, the acetyl groups are attached to the histone tails. So this process loosens chromatin structure and thus initiating transcription. And another process called DNA methylation involves the addition of a methyl group to certain bases in the DNA, and this is usually 2-cytosine. And then this methyl group usually causes the compaction of the chromatin, thus reducing transcription. So far, we have the process of acetylation, which again loosens. And then we have methylation, which again condense. So two things you need to remember to tie those things, to each other. Now, in some species, DNA methylation causes long-term inactivation of genes in cellular differentiation. And then, furthermore, changes made to the chromatin structure are called epigenetic changes, which alter the expressions of phenotypes without changing the genotype or DNA itself. So again, that tells us a lot about how identical twins, they're identical to their genetics, look different. Because again, the manipulation of the chromatin structure causes different expressions of phenotypes without changing that DNA. So, most eukaryotic genes are regulatory sequences. Okay, those are parts of non-coding. Remember, this is a strand being read by RNA polymerase that help regulate transcription by binding to certain proteins. So we're gonna get to know these proteins names. So examples of the regulatory sequences in prokaryotes and eukaryotes, again, in all cells, the promoters. This is on the DNA, which helps to bind RNA polymerase in order for transcription to occur. In prokaryotes, the operators, which bind to the repressors, like we learned in the previous lesson. And then we have the eukaryotic cells. They have something called the enhancers. Again, bind to activator proteins and stimulate the transcription of a gene. So again, the enhancers, the operators, they are on the DNA. So again, these are parts, let's not forget this, parts of the DNA, right, that have specific functions. I'm a very visual person. So again, this is what is being represented visually for eukaryotic genes. So again, this blue thing is all DNA. The segment of DNA that's called the promoter allows for the RNA polymerase to bind to. Then upstream of all of this would be a spot for the enhancer. So again, the proteins that bind to this helps promote the transcription of this segment here. So once transcription occurs, y'all, we do see all of the things being done to make it modified. So let's talk about the promoter region. So the promoter region in eukaryotes contain a common core sequence called the Tata box. So this is a sequence of DNA rich in adenine and thymine base pairs. So the eukaryotic promoters also bind to regulatory proteins called transcription factors, and these help attach to RNA polymerase. Okay, so again, y'all can see the process here. We have the Tata box transcription factors bind to the promoter site, and then the transcription factors help attach the RNA polymerase. Boom, done. So there are two types of transcription factors. The first one is general transcription factors, essential for the transcription of all protein coding genes. Then we have the specific ones that are required for the transcription of only particular genes. With the specific transcription factors, there are two types. The first one activators, which increase expression, and then the second one are repressors, which decrease expression. The activators, they bind to regulatory sequences, again, on the DNA, called enhancers. And then repressors, on the other hand, they bind to regulatory sequences on the DNA, called silencers. So this is a visual of what we saw on the previous slide. So again, the combination of the transcription factors binding to the regulatory regions at any one time determines how much, if any, of the gene product will be produced. So again, combination of transcription factors simply means both the general and the specific. So with the specific, we have in the image activators being represented. The activators are bound to their enhancer regulatory sequences, which is upstream. Notice this is located before the Tata box. So the repressor isn't represented here since it's not bound to this regulatory sequence of DNA called the silencer. Then the activators, along with the general transcription factors, help RNA bind to the promoter region to start transcription. So again, another visual, we see that the enhancer is definitely super duper upstream, okay, of the promoter region. Then DNA bending protein attaches to the segment between the promoter and the activator and enhancer sequences. And we see again group of mediator proteins here, general transcription factors, all combined to help bind RNA polymerase to start transcription. Okay, so gene expression is super complex and involves the coordination of regulatory genes, regulatory sequences, and transcription factors all working together. So, a particular combination of regulatory sequences can activate transcription only when the appropriate activator proteins are there. This will tell us whether a gene is expressed in a cell or not. So let's take a look at a specific example of the albumin gene and the crystalline gene. So again, remember, DNA are in both cells, but the activators are not shown. When we take a look at the liver cell, again, the activators for the albumin gene are there, while the crystalline gene is not, thus it is not expressed. These activators bind and help RNA polymerase undergo transcription. And so thus here, in the lens cells, we see that the activators are not present for the albumin gene. So the albumin is not being expressed, but the crystalline gene is, because again, the activators, proteins, are there to help polymerase bind to the DNA to transcribe it. Unlike the genes of a prokaryotic operon, each individual eukaryotic gene has its own promoter and regulatory sequences. In order to be co-expressed, meaning that they are all being created at once, they have to have the same identical regulatory sequences, and again, the same specific transcription factors. But no, these genes are different. Again, this promotes simultaneous transcription of genes whenever those transcription factors are present in a cell. So we have this primary example here. I have transcription factor A, B, C, and D. They all work together to promote gene X1, X2, and X3. Once the mRNA is created, it is translated into 3 different proteins, but the transcription and translation of these 3 proteins, y'all, happen together. So what we talked about with activators and repressors deals with the regulation of these genes at the transcription level. Now, there's post-transcriptional regulation. And again, this involves alternative RNA splicing and RNA interference. So again, this all relates to regulation after transcription allows a cell to fine-tune gene expression rapidly in response to environmental changes. Without this post-transcriptional regulation, again, adaptation and the response to the environment would be extremely slow. Our first example is alternative RNA splicing. So different mRNAs can be made from one gene. And this is simply done by splicing together different combinations of the exons. So let's take a look at the example. We have the chromosomal DNA. Again, the RNA, or sorry, the pre-mRNA is created. Now, splicing occurs. The order of the exons differs in each of these different final mature mRNA, and thus the proteins are all different. The second example would be RNA interference or RNAi. And RNAi occurs when gene expression is inhibited by small single-stranded RNA molecules called microRNA or miRNA. These microRNAs are transcribed from DNA. Once transcribed, they are folded and processed, then guided to a target mRNA by a protein complex. Then, binding of the miRNAs to the mRNA inhibits translation and causes the mRNA to be degraded. So, to summarize, miRNA marks an mRNA to be degraded. Okay? Translation cannot occur. Boom, done. So this is a quick summary of the differences between regulations in prokaryotes versus regulation in eukaryotes. So for prokaryotes, the regulatory proteins would be the repressors, the activators, okay, then the regulatory sequences, promoters, operators, and then the activator binding sites. Now, we have eukaryotes, and then the regulatory proteins includes the general transcription factors, the specific transcription factors, activators and repressors, the promoters containing that Tata box, and then the enhancers sequence that is upstream of the promoter, and the silencers as well. Okay, y'all, that's it for today. Have a good one.